this point, I would like to introduce Dr. Carey Williams, our presenter for this evening. Dr. Williams earned her doctorate degree with an emphasis on equine nutrition and exercise physiology in June 2003 from Virginia Polytechnic Institute and State University. She holds a master's degree in equine nutrition, also from Virginia Tech, and a bachelor's degree in equine science from Colorado State University. She joined Rutgers University in July 2003 as its Equine Extension Specialist and Associate Director of Outreach for the Equine Science Center, taking an active role in teaching, conducting research, and working with the equine and academic communities to ensure the vitality of the horse industry in New Jersey. Tonight, as a part of the Equine Science Center's Fall 2020 Equine Webinar Series, Dr. Williams will present on her many years of research on oxidative stress in exercising horses and how we can use antioxidant supplementation to help alleviate that stress. Her talk will focus on recommendations for using antibiotics for your athletic horse, no matter what the exercise or the cause of the stress. So at this point, I am honored to turn this over to Dr. Williams.
Excellent. Thank you, Jen, and thank you for the Equine Science Center for inviting me to speak tonight, and thank you all for being here. As I was scrolling through the list of attendees, we got some very familiar names, so thank you for joining us again. And if you're new, or if I might not recognize your name, welcome. I know some of this bits and pieces. If you've been with me over my 18 years, or going on 18 years here at Rutgers, you probably heard bits and pieces of it, but maybe haven't heard it all tied together in a nice little bundle, I guess, of everything I've done in terms of oxidative stress. So we're gonna go ahead and kick it off with just a little bit of, a hot topic. Why do we care? Who says it's really important? Well, whenever you wanna find out about something, what do we do nowadays? Well, we go on Mr. Google, right? Dr. Google, whatever they call it. So I just thought it would be interesting over the years as I give this talk to double check things and see how popular the topic has gotten. And I started doing this in grad school when we had maybe, the 5-digit numbers of how big of a hot topic oxidative stress is. The last time I gave this talk about 5-6 years ago, we were in the low millions. Well, I just changed this yesterday. When you type in oxidative stress into Google, you get 193 million results. So the topic is not going away at all. In fact, it's exploding. So then I'm like, well, let me add horse, because usually there's only, 5,000 or 6,000 when you add horse. Well, now we're getting about 3 million or over 3 million when you say oxidative stress and horse. Okay, so even in the equine field, I was the first one to do anything related to oxidative stress or using antioxidants for that purpose, and it's exploded. So antioxidants alone, drop everything else and just say antioxidant, and it's up to 121 million, and that's up from, like I said, the low millions, kind of the same as oxidative stress. So again, this topic isn't going anywhere. I started working with oxidative stress in the early 2000s, actually the year of 2000. So I've been doing this for about 20 years. It has changed. We've come far, but we still have far to go, because I think with anything dealing with equine nutrition, there's so much more we can learn. So obviously I can't cover all 193 million results. So what I'm going to do, I'm just going to focus on a lot of the highlights from my studies, and then tie in some other laboratories that might have been doing some research as well. I will have different sections of research so we can stop and answer questions as we go, like Jen said. So first, I want to get everybody on the same page, and let's talk about, because we keep saying oxidative stress, oxidative stress. Well, let's get us there. What is it? Well, we have these things called reactive oxygen species, or ROS, as you're going to see it in my next few slides, just for the way of abbreviated, and I'll probably say ROS as well. There are things like hydrogen peroxide. Everybody's heard of hydrogen peroxide. Well, you might not have heard of singlet oxygen. That's another reactive oxygen species. But then you also have free radicals. So free radicals are that term that gets thrown out there all the time. So the free radicals are key because they have this little unpaired electron here. And that unpaired electron really wants to get paired with something else. So it's actually going to pair with some of our healthy tissue. So here's an example, and I don't mean to scare you by going back to biochemistry with some really scary equations. So I simplified it. And all we really need to do is down here, look at oxygen. So in our cells, we have oxygen that basically just goes to form two waters, okay? And in the process, we're using some hydrogens and some carbon dioxide. But this isn't a clean reaction. This isn't a 100% reaction. What happens is it actually causes some of these reactive oxygen species to spin off. And it's in the realm of about 2 to 4% of the reaction actually does cause that reactive oxygen species, or the ROS. So what happens is when we have these ROS production, the more and more and more that accumulates in the system, it can actually overwhelm the capacity of the antioxidant system. If we're healthy and we have plenty of antioxidants in the body, we'll never know we have oxidative stress. You might be sitting there right now with some ROS being produced, but we have enough antioxidants and don't need to worry about it. But it's when that these start to overwhelm the system that you'll compromise the homeostatic environment, and you can degrade some protein, DNA, and polyunsaturated fatty acids. Now, I do want to say some oxidative stress is a good thing. Throughout this talk, you're going to hear me talking about it being bad, okay? And that's because I'm getting to the point of my talk and why we're using antioxidants. However, that's not always the case. We do need ROS. We do need some of these free radicals to help us fight off bacterial infections, to help when we're sick, basically, and to help with our immune system as well. But you just need it to be at a decent level. You don't want it too high or too low. You need kind of that optimal range, okay? So what has oxidative stress been associated with? Well, particularly in the horse, we found that exercise, as the horse gets older or during aging, and then in humans, a bunch of diseases have been led back to some form of reactive oxygen species, like diabetes, Alzheimer's, various neurodegenerative diseases, and cataract development, just to name a few. There's many more of the last recent years that also have been linked back to excess ROS. So ROS, like I mentioned, can be combated, or the defense mechanism against these is our antioxidants. And there are many of them. There are an awful lot. Some that might be more familiar to you are things like vitamins, like vitamin E, C, and A are a few that I'm especially going to talk about vitamin E and C because I've done more work with those. Various enzymes, you'll hear me mention glutathione peroxidase, which is GPS, and superoxide dismutase, which is SOD. And then there's a bunch of others, things like glutathione in general, lipoic acid, and CoQ10. And I'm going to touch on a lot of these, but again, I'm not going to be able to cover the whole body of research because, like I said, there's 121 million Google results on antioxidants. So here's just a quick little schematic, a really brief little, very simplified schematic of roughly what's going on in our cells when we have antioxidants trying to decrease the ROS production. So what we're looking at here is a magnified version of a cell membrane. So here's the cell membrane. We have everything outside of the cell and everything inside of the cell, okay? So all of these little things are polyunsaturated fatty acids. They're individual PUFAs, okay, as we call them. In between the PUFAs are vitamin E molecules. So you see all the vitamin E molecules are in pink. So what happens if we have a free radical interact or try to destroy one of these PUFAs? Instead of having a chain reaction destroy the whole membrane, vitamin E is going to stop it. It's going to be like hitting a brick wall. And then it recycles that ROS into something that's non-reactive, like water. However, in the process of doing that, vitamin E forms its own radical. Granted, it's not a very reactive radical. It's a fairly benign radical. But in essence, it can't do any more than it's already done. It basically stops. Oops. Sorry, hit another button. So we also have vitamin C. So vitamin C doesn't incorporate into membranes because it's water soluble. So it's out in the extracellular or intercellular space, but it's going to do the same thing. It's going to render that free radical inactive, turning it into a harmless molecule. And again, vitamin C is therefore inactive. But the fun thing about the vitamin C molecules and all antioxidants in general, they love to work together and in combination with each other. So vitamin C can also recycle vitamin E so that it can continue its action. It's not like you throw it in the trash and you get a new one. You're actually able to recycle it and use it again, kind of like a rechargeable battery. And all of these, so vitamin C also has its antioxidants that recycle it, and so on and so forth. I have another slide in a little bit that actually ties together a lot more of the antioxidants than just vitamin E and C. So as we go through some of my research, you'll see I never actually measure the ROS. And that's because what we just saw on the other slide, they're always changing so rapidly. They're either increasing, decreasing, moving around. You can never get a snapshot of how many ROS are in the system. So what we have to do is look at footprints that it leaves behind, or intermediates or end products from an excess or from the ROS, things that they leave behind. And some of these are things like the LPO is a lipid hydroperoxide molecule. So it's after the lipids have been oxidized, you'll be able to measure that. It's the same, these T-bars, and in the last 20 years, I still haven't been able to pronounce this word right, but it sounds like thiobuturic acid reactive substances, and that is the only time you're going to get me to say that. And MDA is malonyl-dialdehyde. Again, these are all end products of the oxidation left behind by the ROS. So you'll hear these terms a lot more. Same with these F2 isoprostanes, same thing. There's also some redox reactions between oxidized and reduced molecules as well. So you're going to hear these a lot more as we go through some of the research. So I know that was a very simplified version as to what happens with ROS and what the antioxidants are. I could probably talk for another 3 hours on all the details, but I really want to get into some of the research so that you see what's really been done in horses. Because again, there has been a lot done. It's just, you know, a lot more isn't done in humans, let's put it that way. So I've got 3 main studies that we're going to talk about first that really are in the realm of endurance exercise, because that's where I started when I was a graduate student. When I was doing my PhD, I worked in Virginia with the endurance horse. And the reason why the endurance horse made such a great model for oxidative stress is because as they're exercising, they use more of that, the slow twitch muscle, and the slow twitch muscles use a lot more of the oxidative enzymes. So any long-term endurance type exercise has the capacity to have higher levels of oxidative stress. So we've got 3 studies that we're going to go through here, and I'm just going to jump right in with a brief little introduction to about vitamin E, because that's one of the things we're looking at a little closer with these studies. So here's this T-bars again. Told you I was never going to say the whole word again, but there's been studies way back in the 90s, this was long before I was working on it, that did look at a little bit of the T-bars. They didn't really understand them a lot, but they saw that, hey, these increase after exercise. And there's more increase in them when horses with low vitamin E. So that's what that study concluded. Another study back in the 90s as well also concluded that, well, if you're just sub-maximally exercising, meaning, you know, you might be going out for a light trail ride with your horse, doing a little bit of trot, a little bit of canter, but it's not very intense. So this doesn't affect the vitamin E in the system at all. However, the more intense the exercise gets, the thought was back in the 90s that maybe they're going to need more vitamin E to help decrease some of this exercise. So back in the early 2000s, she was a colleague of mine at Virginia Tech, was up in Connecticut where they have an awful lot of polo ponies. So when she was looking at exercise, they used polo ponies. Some of them didn't work very hard. Other ones worked very intensely. So she had a nice gamut of horses that were either like the lightly ridden horses and the intensely ridden horses. And she looked at vitamin E and vitamin C in combination with each other and found that the horses that were exercising harder had higher levels of the reactive oxygen species, or the lipid peroxides left behind, but they also had higher alpha tocopherol and ascorbate, or they needed more, and they had a lot more in their system as well. So they were using a lot of it, and they were reacting together back and forth, like I showed you in that one slide, with the lipid peroxide membranes. So that brings me to some of the start of some of my research in graduate school with these endurance horses. First, we asked them what their diet was like. These are highly competitive endurance horses that are going 50 to 100 miles in a ride, taking 8, 12, even 24 hours in some of their competitions. And we found in each one of these dots on this graph represents a horse. Anywhere from 1,150 up to almost 5,000 IU per day is what these horses were getting supplemented, or what they were eating in their diet. We averaged, or we estimated the amount of pasture intake, but we also calculated it in their diet and in their supplement. So you may be asking yourself, well, I have no idea what this means. Like, what does an average horse get? Well, if you think about it, the maintenance horse, so a horse just standing out in the field, 1,000-pound animal. I do easy math, I'll admit. All my horses are 1,000 pounds, because that makes it much easier. But that means about a 1,000-pound animal at maintenance needs about 500 IU per day. So it's not a whole lot, but it doubles for any sort of production, whether it's heavy exercise, a brood mare, a lactating mare, a growing horse. That amount doubles. So you're looking at, for maximal exercise, you should be about this 1,100 mark. So that's actually the bare minimum that they should be getting. All the way up to this 4,700 is what some of these horses were getting. So what does that mean? Well, this is just the start, like I said, of some of the research we were doing. What we did also find in this little study, this was just kind of a survey study looking at these horses. We calculated, or we looked at in their blood, different levels of enzymes and the alpha tocopherol itself, which is that vitamin E molecule. These CKs and ASTs, if you have ever had a horse that ties up, or you go to the vet and your vet says we need to look at the muscle enzymes, that's what these are. It's creatine kinase and aspartate aminotransferase. And both of those are supposed to be in muscle cells. They're muscle enzymes that help with muscle metabolism, and they need to stay in the muscle. The problem is, after exercise, as our membranes start to get a little softer and a little more permeable, they leak out into the bloodstream. So the neat thing is, we don't need to be taking muscle samples to see how much enzyme is in there. We can take a blood sample, and if there's really high levels of these enzymes, we know there's been some sort of membrane leakage or membrane permeability increase in these muscle cells. So with an increase in vitamin E intake, we have decreased the amount of these muscle enzymes, or therefore we can theorize that we've decreased the amount of membrane permeability or membrane damage to these muscle cells. We've also increased the level of alpha tocopherol. So feed vitamin E, you get more vitamin E in the system. So what we did with this first study, we had a research ride at Virginia Tech. This was a race, a sanctioned race that horses came to and actually did research for us. So they were ridden by competitive riders. These were competitive horses. About 3 weeks prior to the race, we sent them one of two treatments. They were either on 5,000 IU, because remember, we saw that this is about what a lot of the horses at the highest level were getting fed, or they were getting E and C. So they had 5,000 vitamin E with 7 grams of vitamin C, which is about that level of vitamin C you would want to give for a horse that's undergoing a lot of stress. Now, you might say, well, where's the control group? Well, because we were using other people's horses, we didn't have a control group. So keep this in mind as we go through some of my other research. We had about a 74% completion rate. That's good. We only looked at what was going on with the finished horses now, and you'll see why in a little bit. We did find that a lot increased after exercise. So we looked at the entire 80 kilometers. This was an 80-kilometer endurance ride. we looked at, or 50 miles, we looked at all of it. We looked at temperature, heart rate, these muscle enzymes again. We looked at the lipid peroxides, and then our glutathione and glutathione peroxidase, which are those antioxidants and antioxidant enzymes. So unfortunately, we didn't have a whole lot of treatment differences between the E and the E and C group other than ascorbate. So ascorbate is what we're looking at here. The E and C group is in the blue, and the vitamin E alone is in the pink. And here on the bottom, and this is, most of my graphs will look like this. On the bottom, we'll have the mileage, basically, that the horse went, or the sample times we took. We took it before, 21 kilometers, 56 kilometers, 80 kilometers, and then about an hour of recovery. And you see that the horses that were given the vitamin C, that's higher vitamin C. So you might think, all right, well, that's pretty obvious. Well, not necessarily, because there's been other studies and other labs that have actually found that vitamin C can decrease during exercise. Well, let me just go back up a little bit. There was no decrease during exercise here. The vitamin C group maintained, and the vitamin E group increased. So something different's going on with supplemented horses. And again, this study, the Hargrave study, didn't have supplementation as a factor. They've also found the same thing in sled dogs. Sled dogs is another highly competitive, highly oxidative type of exercise in these animals. They found that plasma vitamin C or ascorbate decreased during the race and throughout the season. However, when these animals were given vitamin C or were supplemented with vitamin C, they actually prevented this decline, and they maintained or they increased, which is similar to what we saw in my study. However, these sled dogs were given about 3 times that amount. And this is on a metabolic body weight equivalent. So yeah, they're dogs, they're smaller, but we put it kind of on the same level. So that was what was interesting there, is that, yeah, maybe some of these more competitive horses will need something more than just vitamin E. Maybe they'll also need vitamin C to help with this antioxidant capacity. So what we did see with this study is that, yes, the lipid peroxides increased, and yes, also the muscle enzymes increased. So that is indicative of horses undergoing oxidative stress. However, was antioxidant supplementation really necessary? Well, we can't really say that yet, right? Because we didn't have a control group. So again, keep that in mind. So now what about horses that didn't finish the race? Because these are all of our finishers. What happened with them, and was there any difference with them? Well, we had another study, and this was actually a different year, where we had, still some very competitive horses and riders. This time we weren't giving them any supplements. We just did nutritional surveys and took blood samples. And we had 24 finish the race, 16 not finish. And what we found is that the non-finishers are in the blue, the finishers are in the pink. This is that creatine kinase, that muscle enzyme. Even if they didn't tie up, so tying up is a condition that happens quite frequently, actually, in endurance horses. And it is a big thing for these endurance riders to try to prevent that. So what happens here is that the finishers, regardless of how they didn't finish the race or why they didn't finish the race, had much higher levels of creatine kinase. And you'll see my scale here is a log scale. And let me tell you, we had the low of about 100 IU per liter up to in the hundreds of thousands. So I'm not able to put this on the same chart unless I did a log transformation. So that's what that is. But you can see, yeah, why don't we have 80 kilometers for the non-finishers? Well, that's because they didn't make it that far. So we didn't get a final, but we did get the recovery sample. And there's still, all these stars mean that there are differences between the finishers and the non-finishers. So where does that leave us? Well, that left us, especially as I was completing some of my PhD work, to ask myself, well, now I really want to know what's going to happen to these horses that aren't fed any antioxidants above that basal level of the diet. So the only way we were able to do that is to get our own horses. And in order to do that, we had to get a treadmill to be able to exercise our own horses. So we did that with, these were endurance trained horses. Actually, myself and my colleague, Dr. Tanya Hess, we rode the horses. We trained them just like we would for a competition. She is an avid endurance rider. It was a lot of fun. and a lot of work. And we had these 3 groups. We did a vitamin E group and we did a control group, which only had what was in their hay and their little bit of grain. And then we also had a lipoic acid group. So hold on just 1 minute. If you've never heard of lipoic acid, I'll tell you about it in just a minute. So we tested 1 horse per day because they ran 50 kilometers on a treadmill. It took us about 5 hours per horse. So what's lipoic acid? Well, I was fascinated by this molecule as a graduate student because I started reading on all the cool things it did in humans. It was doing all sorts of fun stuff from, and I'll pull all these in here, helping humans with neurologic symptoms in neuropathies of diabetes, patients with Alzheimer's. It was found to be fat soluble and water soluble. So that's because of its structure with its 8 carbons and its 5 carbon tail. It was actually discovered not all that long ago in 1951. So not all that old of a molecule. They did a lot with rats and found that with exercise, it does decrease oxidative stress and increases other antioxidants. So let's use it in horses. Why not, right? So here's that other slide that shows the massive interaction of antioxidants. And I'm pulling together more than just E and C. And the one we've thrown out there a lot is glutathione. So that's this GSH. Glutathione, it can be reduced, and its GSSH is its reduced form. Now, we do need vitamin C in order to recycle glutathione as well as vitamin E. So vitamin C, there's its reduced form, vitamin E radical in its vitamin E form. So glutathione, vitamin C, and vitamin E are all directly related and do recycle and interact with each other. What also happens is lipoic acid is tied in directly to vitamin C and indirectly to glutathione through cysteine and cysteine molecules. So this is even just, I actually took this or adapted this from an article that was written in the 2000s on humans and antioxidants, and I cut out about half of all the interactions here just to make it a little clearer on what we're doing here. So the interactions here are far beyond what this is, but this is just illustrating that the interactions are pretty profound. And actually, I just glanced over to the chat, and Maggie asked, could lipoic acid help Alzheimer's patients? And absolutely yes. So when I was first using this molecule in horses, they were actively using it in trials and treatments with patients in Europe. Granted, it wasn't approved for patients in the US. We have a little bit stricter policy on some of that. But in Europe, they were successfully using this in a lot of patients, both diabetic, neuropathy patients, and Alzheimer's patients. So here's what this study looked like, because remember I said we exercised horses for 55 kilometers on a treadmill. So what we did is we had loops, just like in a simulated endurance ride. Anybody's done even a competitive trail ride, you know that there's checks, there's little vet checks in between. So what we did is we had loops, or we had vet checks. So we had a 20-mile loop, and then we had a 40-mile loop, and then we had a 20 and a 20 and a 15. And where we took blood samples before they started exercising, at the peak of their exercise. Remember, we're on horses on a treadmill, and that's the picture you saw with my colleague taking blood samples as they're galloping along. And that's, as many of you may have seen, Dr. McKeever and the Equine Science Center's treadmill demos. That's the beauty of doing things even here at Rutgers. And then we took samples during recovery period. We did, this is it's just 60, but it was an hour recovery. So you'll see my graphs are going to look exactly like this with all of the mileage down at the bottom. And then you'll have the pre's and the post there. So don't think you're seeing things. You don't need to rub your eyes. I haven't given you the results yet. This is actually, I'm going to pull them in. It gets really complicated because there's so many samples, but this is that muscle enzyme. This is that muscle enzyme that you want to stay in the muscle, but we're going to measure it in the blood. And here are the horses that were given the vitamin E. Here are the horses that were given the lipoic acid. Okay, you see there's a little bit of error and some small differences, but there really wasn't extreme differences here. Where there does get to be a lot of the differences is these horses that did not receive any supplementation. And yes, there's big variability as well, and on average, these horses had much higher levels by the time they were at the end of the race and into recovery if they did not receive any antioxidant supplementation, whether it was vitamin E or lipoic acid. And I also see what speed were they at. And actually, good question. I did kind of pass that up. This was based on heart rate max. So for the most part, when they were at 30, they were 30% of their max. They were just walking. Once they got up to about 40 to 50, they were at a good trot clip. And then that 60%, they were galloping along pretty good. Okay, so that's how it went. They walked, they trotted, they cantered, they trotted, they walked, they stopped. They walked, they trotted, they cantered. So that was those loops. Good question. Sorry, I skipped over that. So the next graph, this is going to be the vitamin E in the plasma. So the beta carotene, or the alpha tocopherol in the system. So this is going to show you how these antioxidants all collaborate together. We have the vitamin E group, which you expect to be high because they were fed vitamin E, right? However, here's the lipoic acid group. Yeah, it's a little bit lower, but the interesting thing is it's still much higher than that control group, which didn't get any antioxidants. So this again goes back to that slide where I showed so many of the interactions together. We also looked at something called apoptosis, or programmed cell death. There has been a theory that programmed cell death will occur after intense exercise, especially endurance exercise, but we didn't see any increases with either of the supplemented groups, lipoic acid or vitamin E. However, here's what we have for those control horses. So they have up to around 30% on average of this apoptosis. The interesting thing was there was 3 horses that ran on days which were much, much hotter. This actually happened in April in Virginia. So the temperatures usually are in the 60s, but we had 80 to 90 degree days. And the horses were just through the roof in terms of their apoptosis, these particular horses. And wouldn't you know it, I did not plan this. These were randomly assigned. There was 1 horse from each treatment group. So treatment did not have an effect when they were running on very hot days. So just some interesting stuff. For the sake of time, I've been watching the chat. So for the sake of time, I'm actually not going to stop for questions. I'm going to keep going, but I do have the chat open right next to me. So please go ahead and keep asking if you need me to stop in there. So now, okay, let's leave my graduate work behind. That was too long ago. Now I come to Rutgers. What horse do we have? Everybody should know that, right? We've got standardbreds. What do standardbreds do? They trot like the wind. They are some amazing animals, and I've had so much fun working with them over the years. So I switched and said, okay, I worked with endurance. Now I want to see what's really happening with these horses. Can we do the same thing with vitamin E? Can we do the same thing with some of these other antioxidants and nutraceuticals? So the first study was pretty cut and dry. I still wanted to use that 5,000 IU per day of the alpha tocopherol. But I started having vets ask me, especially vets at the track, what happens if I double that? Is it any better? Can I go up to 10,000 IU per day? Well, we did basically the same sort of study, only we looked at an interval test. So we had horses gallop and then walk, and then gallop, then walk, and gallop, then walk. And yes, some of my standardbreds did gallop on the treadmill. So this is what it looked like. Actually, we had trot, canter, or gallop. They were at 100% of their heart rate max. So they were going at a good clip. And we took samples at those 4 time points. And that's what you're going to see down on the bottom of my graph here. We had the pre-sample, the trot, the canter, and the walk, and then some recovery times. So we have, the colors are different here than they were on the last slide. Sorry, I should have changed them. But now we have this blue is the control group. The red is that group that got the 5,000 IU, and the yellow is the 10,000 IU. So these are those muscle enzymes again. You can see the differences here. We still have increase after exercise with the creatine kinase, but with that control group, it's even higher than we had with both of the treatment groups. Both of the treatment groups didn't differ at all. Alpha tocopherol in the system, to my surprise, we didn't have any differences until this very last time sample. Even before, we had a little bit higher numbers, but remember with some of my numbers in the endurance horses, we were up around 5 to 6. So we would have been way up in this area with some of my endurance horses, but we didn't quite get there with the standardbreds until the very end of the race with this very high group. So, you know, what's the sense in feeding 10,000 IU? Right now, I haven't shown that it's any different. However, there is a difference, but it might not be a good difference. Because what we showed is that, and remember, the red is that 5,000, the yellow is the 10,000. And what we're looking at here is beta carotene in the system. Beta carotene is the vitamin precursor to vitamin A, but it's also an antioxidant. We are actually finding that as you supplement excesses over what we've been doing in the past, so these 10,000 IU per day, we've got much lower levels of the beta carotene. And this is much lower than the 5,000 IU group. So what's happening here is you're starting to interfere with the carriers. So the alpha tocopherol and the beta carotene fight for the same carrier to take it into the system. So what's happening here is the excess vitamin E is winning, basically. So unless you have your horses on a lot of green, fresh pasture that has a lot of beta carotene in it, you might be starting to run into problems. Now, these weren't deficient, but again, depending on their normal diet, they might start to have problems. So that's the only reason why I would caution using the 10,000. Plus, if you're not having any extra added benefit, you're really just wasting your money. We've also worked with superoxide dismutase. One of my graduate students did some work on this. And here we did a repeated sprint test where we did, this is where we did basically walk, gallop, walk, gallop, walk, gallop, to try to get the horse fatigued as possible. And we looked at inflammation as well as some antioxidants. And the reason why we did this is because I also worked with a colleague of mine in the exercise science department with soccer players. And yes, we used the Rutgers soccer team, and that was a lot of fun to help with. And they found that when they were given superoxide dismutase, these athletes after, and this was after a full season of training, had much lower levels. So we're looking at this graph here, much lower levels of these isoprostanes, which is the lipid peroxides. Okay, so these lipid peroxides were much lower in soccer players after a full season of training. So this was a long training period. They also found in the football players, and yes, we used football players in this study, Rutgers football players, that were supplemented over 7 weeks. Here they did a pre, a post, and a recovery sample after they were on, they did a bike test, so a maximal bike test. And here what we're looking at is this solid yellow line at the bottom, so this line right here, are the only athletes that were exercised after the season and had lower lipid peroxides in the system. So again, SOD looks great in humans, so let's try it in horses. And I see Maggie says, would they have peed it out? Would they pee it out and get more if they get more than they need? So I'm assuming you're talking about vitamin E. And Maggie, vitamin E is a fat-soluble vitamin, so it does not get excreted in the urine. Vitamin C, on the other hand, can, but not vitamin E. Vitamin E will get stored in the body and stored in the muscle cells, stored in the fat cells as well. So what we found with the horses, and you see there's not 2 bars here like there has been in everything else, or 3 bars for that matter, and that's because there were 0 benefits or differences at all with the superoxide dismutase treatment. We either had it or we didn't, and there was no differences. The only thing we found was with these cytokine transcripts, so this was inflammation in the system. It either peaked after exercise, so we knew the test was working. It peaked either 30 minutes or 2 hours after exercise, but there were no differences in the SOD. So here is actually superoxide dismutase activity in the red blood cell, because SOD is actually housed in the red blood cell. There was no differences other than the fact that it does increase by the end of exercise. So yes, it is compensating for an increase in oxidative stress, because we did see that with the increase in the lipid peroxides and the increase in the creatine kinase, but we didn't see any differences with the supplementation. But if you remember from my human athletes, what were they doing? What were the human athletes doing? They were Rutgers athletes, right? They were soccer players. They were football players. They were training. It was all during their training, from pre-training to post-training. They were exercising intensely, getting ready for their season. What were our horses doing? Well, I kind of skipped over that at the beginning, but I can tell you they were good old pasture pets. They ran one test before we started the trial and one test after. And that's it. They got to roam around the pasture and do nothing. So what would have been more interesting is if we actually would have had a training protocol that we had these horses under that simulated more so the human athletes than the study that we did, which was basically with couch potatoes. I piggybacked onto a study by Dr. McKeever. He was looking at some nutraceuticals and the inflammation, but I really wanted to look at the antioxidant status and some oxidative stress and how these nutraceuticals would affect it. We had one study using, here's our lovely control groups, but they were ginger or cranberry or black tea or orange peel. And unfortunately, I didn't find any differences with these nutraceuticals after one megadose. So this would have been like if you were to chug a concentrate of, a superfood. So it wasn't over a long period of time. It was just one dose. But we really didn't see anything other than, we measured retinol and it was decreased in supplemented horses, which orange peel supplemented. So that was interesting. But really, it would have been interesting to see if there was any long-term effects, if we were to give any of these nutraceuticals long-term. So the studies by Dr. McKeever's research, or graduate students did find that, yes, they did have some anti-inflammatory properties. The anti-inflammatory or the inflammatory markers decreased with black tea and cranberry. So those were kind of the biggest ones. The orange peel and the ginger did decrease recovery time in these horses, but ginger was very caustic to the system and actually increased the creatine kinase. So all of these nutraceuticals were produced by the Rutgers Food Science Department. So it was really interesting because they did actually take them from there. And my same colleague, who I showed you the results from with his SOD studies, actually used black tea and cranberry in the human athletes as well. So we were kind of the stepping stone to that. So I am just going to close with a couple more random studies, and I say random because we didn't actually supplement anything. We were just looking at oxidative stress. I had heard so much about this inflammaging and ROS, free radical theory of aging, and all of this stuff about aging. And I really wanted to find out what happens with old horses and are young horses any different. And then I also wanted to take it to competition. So we have some 3-day event horses we've researched as well. So first study, we had young horses, which young, I mean about 12 years of age. And then we had old horses, so about 22 years of age. And they did a exercise test. They did train over 8 weeks. So they were on an exercise or training and then another exercise test. Unfortunately, I saw no effect of oxidative stress or the antioxidants with the old horses. And I was disappointed. I was really hoping this was going to be a breakthrough. But what we did see is an effect of training. We saw that with training, and it was only after, so this is after exercise, and we're looking at glutathione here, we saw that there was an increase. And we thought that, okay, this is pre-training. These horses have much more glutathione because it's needed more in the system. They are producing more ROS. They've not exercised before. They just got off the couch. Now they need to exercise more, so they need more of these antioxidants to combat the oxidative stress. However, with exercise, they're fairly fit, and they don't really need to prime the system with extra antioxidants because their oxidative stress is lower. Where we did see the difference with age, however, is in that apoptosis again, that programmed cell death. My young horses stayed very low, about 10%, until after exercise, they increased a little bit. However, these old horses started high and ended fairly high. Granted, the last sample, they weren't different because there's such a large error range. But during and at the max of the exercise, there was much higher levels of apoptosis and this is white blood cell apoptosis. I keep saying system, but in the white blood cells in the older horses. So same with the differences we saw with the hot weather. So
think in your brain, do I have an older horse? And do I exercise them when it's really hot? Because those two things combined are both detrimental to the white blood cell apoptosis. So you might not want to combine those two factors. My next study, and this was actually with my other graduate student, Dr. Danielle Smarsh, she looked at young horses now. I wanted to know, or we wanted to know, what happens when you take these yearlings, and I'm saying long yearlings, 16 to 18 months, and these horses, I will say, were barely halter broke when we got them, and compare them to that same moderate aged or middle-aged horse, 10 to 15 years. We had trained and untrained this time. We didn't train all of them. We ran them on the treadmill. We ran another treadmill test. We did 8 weeks of training, which we also did some high intensity training on the treadmill during this time. And we did another exercise test. So what you're going to see, I'm only going to highlight from the first exercise test for this massive amount of data, which I know it looks confusing. I'm going to give you only what you need. And that's basically that we're looking at MDA, so all of those lipid peroxides in the system, in the muscle. So we did take muscle samples. I want to know what was going on with the young horse's muscles. And then the MDA in the plasma. So look up here. Muscle in yearlings is higher than the mares. Plasma in mares is higher than the yearlings. So it flips. And we theorize here because young horses have so much metabolism going on in their muscles. They're little guys. They are growing rapidly, especially in their muscle. They've never exercised before. They were totally novel to exercise at this point. So the extra muscle metabolism is actually creating higher oxidation in the yearlings, and antioxidants antioxidants in the blood are kind of keeping things in check. Now, what happens? And I see I got a question coming in, but hang on just 1 minute here. Because here's what happened. Here is all of our mares before training. So this is what I just showed you there. And this is, again, this is just the plasma MDA. And here are the mares after training. So with our older mares, they do get a decrease in the oxidative stress in their system after training. So that goes back to what I was showing you in the last old horse study. Training really does benefit our older horses. However, with the yearlings, they are always low. Even before training, they start off low, they end low. These yearlings are always at the same standpoint that our older horses are after training. So we did some other things like muscle nitric oxide and muscle glutathione. And here again, because the young horses have so much muscle metabolism going on, they're higher in the yearlings, both with the nitric oxide and the glutathione. As animals get older in general, there is less nitric oxide available, and the glutathione is just helping fight more of that oxidative stress going on. So this all kind of does make sense. But what does it all mean in terms of the young horses? Well, age does play a huge role. These yearlings have lower oxidative stress before they even start training, and they're better able to cope with the acute exercise we ask them to do at such a young age. But training does benefit mares and older horses. So youth is a good defense, but if you don't have youth, you should really keep training. I know I'm running out of time, and I want to leave a little bit of time for questions. I had a great time doing some studies at the Jersey Fresh International 3-day event at the Horse Park back in 2006 and 2007. I have 4 studies published from this work, and it was a great experience for all my students. We even collaborated with University of Maryland here. We had riders participate in the study. We had 50% of the riders that competed at very high levels. This 2-star, 3-star, this is just underneath Olympic level. So we were the first report of any inflammation, antioxidant status, or oxidative stress in competitive event horses. Other studies have found that, yeah, CKs, ASTs, lactates, they all increase. And environment does play a role. But what we showed is that through a series nutrition surveys. I mean, we really asked a lot of questions of these riders, and we got full dietary protocols and surveys done. They were on average supplemented with 2 to 4 times the amount of recommended minerals, and these are minerals. Calcium, phosphorus, potassium, and magnesium were the 4 main ones that we looked at. They were also high in vitamin E, but most of them were no higher than the 5,000 IU. We also found that over time of the competition, so from before, during, and after the competition, they had a decrease in the plasma nitric oxide in this inflammatory parameter, which is TNF-alpha. That was a marker of inflammation. And this negatively correlated with vitamin E. So the higher the vitamin E level in these horses, the lower the nitric oxide and the lower the TNF-alpha, the lower the inflammation. We also found that horses that were competing at the higher level had higher levels of, or lower levels, sorry, of the glutathione. So the 2 stars had higher than the 3 stars, which meant that they actually needed more of the antioxidants. They had more of the antioxidants in their system, trying to clear out the oxidative stress. So it could mean that their level of fitness had something to do with it, because a lot of these 2-star horses were new to the level, were younger, more inexperienced riders. A lot of these 3-star horses, and actually one of the pictures is Teddy O'Connor. Some of you might remember, yep, there's Teddy O'Connor, the Wonder Pony back in the day. So we did have some pretty major horses on the study. So you know, I've been talking a lot, been throwing out a lot of numbers. So what really does it all mean when it's all said and done? Well, again, there needs to be a lot more research done, I think, to really come to a concrete conclusion. And to tell you the truth, I don't know that we ever will, because they still haven't in humans. But we can say that, yes, horses undergo oxidative stress with exercise, with endurance exercise, with intense exercise, and those on the treadmill. To the extent it does vary, it depends on the ambient temperature, as we saw. It depends on the horse's age and fitness, and the work level and intensity. Can we say that antioxidants are beneficial? Well, I've seen enough positive reports, especially with the methyl enzymes alone, if you leave everything else out, that yes, I believe that we can provide better health and welfare to our athletes by feeding them higher than recommended levels of these antioxidants, especially when they're competing heavily or might not be on pasture. And that is why I have decided to switch my gears and start to look at, with my graduate student Jennifer Weiner, more of the benefits of pasture and how horses metabolize pasture more, because I'm a firm believer that pasture is probably that magic drug that will cure all. So before I start to take, I'll look back at the chat questions, and then I'll take some other questions. This was, like I said, you know, 20 years worth of work. I had a lot of fun doing it, and, you know, have actually given talks similar to this or different types of talks like this all over the world. So it's really been a lot of fun. But all of the faculty, staff, my 2 graduate students who did the bulk of this work, all my undergrads here, different years of undergrads, you know, it was an awful lot of fun. So with that, I hope I didn't get too far ahead of myself, so I have to go back too much and answer questions. But I saw one was dealing with selenium, and I kind of wanted to save that anyway. So Gabriel asked, where does selenium fall in, I guess, and where included with vitamin E and a lot of supplements? So yes, selenium does fall into play. It is a cofactor for glutathione peroxidase. So selenium incorporates itself into glutathione peroxidase, which helps recycle all of these antioxidants. So remember, like I said and showed you in that graph, they all work together in combination. Well, selenium, when added to vitamin E, will help potentiate that activity. So instead of just having a 1-to-1, you'll have like a 1-to-10 ratio. You'll increase the activity or the benefit of vitamin E that more. However, the thing to be careful with that is when you're giving 5,000 IU, most of the supplements that have selenium in it are only at 1,000 IU of vitamin E. So you just really have to be careful, because you wouldn't want to give 5 scoops of that if it has selenium in it, because selenium can be toxic. So just be careful. Is C a logical consideration for supplementation through digestion or adequate in most grazing horses? Yes, vitamin C, and I should have backed up again. I could still be talking about this tomorrow if you let me. Vitamin C is not typically needed in most horses because unlike humans, horses' liver makes the vitamin C. They typically make plenty of vitamin C. That's why there's no dietary requirement for vitamin C for grazing horses or for the average horse. However, horses that are stressed are using a lot more vitamin C. So there's actually a requirement for vitamin C in pregnant horses, lactating horses, exercising horses, because the liver might not be able to keep up with as much as they need. So you just need to be careful if you're supplementing vitamin C, make sure when you remove that vitamin C supplement that you do so gradually because the liver needs time to kind of kick back into gear to make the amount that it needs. These are great questions. Let me take a sip while I read Maggie's next question. That the higher percent in mares, a higher percent of mares tie up than geldings and stallions. Could mares get more supplement? Absolutely. So, you know, I know that on the track, it's typical that a filly will be, and I've heard the term tie-up filly. I have one, an off-the-track thoroughbred. It was a tie-up filly. So I know all about that. It's typical that, or it's thought that maybe some of their hormones also come into play here. And there's not been a whole lot of research done with that. But also typically the more nervous horses. So if you look at ones that are more nervous, typically the fillies, they're going to have more incidence of tying up or more severity even of tying up. And yeah, they can get supplemented more. I was working with some trainers on the track a few years ago and suggested doing the 5,000 IU of vitamin E. And they saw a decrease in the amount of tying up they had. Granted, we also did change some of their management, which, you know, a lot of them were managed pretty well. They tried not to give them a day off. When they were off, they were turned out, that sort of thing, because that does lead to it as well. But yeah, I would say that the mares can get more than any of the stallions or geldings, or just in general, horses that tie up can get more than horses that don't. What kind of special care should we be taking with warm blood yearlings on limited pasture? And that's a great question, Katie. So warm blood yearlings are, you know, they've got a whole bunch of growth things they're dealing with too, because a lot of them are very rapidly growing, not just bone growth, but also muscle growth. So if they're on limited pasture, then they should be at least on a vitamin supplement of some sort. I don't know what the nutritional program is for your yearling, but they should be on a really high quality hay. Even a legume mix would be ideal because especially if they're yearlings, they're going to need more protein. But they also should probably be on a vitamin supplement. And most of the vitamin supplements are going to have a higher level of the antioxidant vitamins in it. You could look for a specific antioxidant supplement. Those will be a little more expensive, but it'll include things like the CoQ10, glutathione peroxidase, and those sorts of things. But even just a vitamin supplement should have enough. Or you can just go with the straight vitamin E. Really, you can go with a, you know, even just probably a 2,500 IU vitamin E. You can look for the natural form, and that'll really help as well. It'll help with their growth. It'll help when they start training as well, especially if they're on limited pasture. And again, I'm such a firm believer in pasture. I never had any of my horses supplemented when they were on pasture. Wouldn't you know it, my tie-up filly 10 years later took her off of pasture, exercised her really hard right out of a stall, and she started tying up again. So yeah, firm believer of pasture, not just for its nutrients, but for the exercise capabilities as well. So I haven't seen any more in the chat, and I know I'm at my time frame, but I'm happy to stay more if there's more questions along these lines. My email is up here. You can always email me. Oh, it's not up anymore, but you can email me. Feel free if you have any further questions, just say you listened to my talk and you had some more questions. I'm happy to answer them. And I will turn it back to our host.
Well, Thank you, Dr. Williams. We can put Dr. Williams' email into the chat. I'm sorry, I closed it out and went to our main slide here before she had a chance to slide that in there, but we'll make sure we get that into the chat for you. Thank you so much, Dr. Williams, for that great talk. I see a lot of thank yous in the chat as well for you. And thank you to all of our participants for logging on and joining us tonight for our talk and for our Fall 2020 Equine Webinar Series. We invite you to come back and join us in 2 weeks when Ed Wingren will be discussing issues facing the New Jersey horse industry to ensure sustainability. If you haven't registered yet, even if you didn't register for that webinar when you registered for tonight's webinar, you can always go back and register at any time. And so again, if you have any questions for us, you can put it in the chat. Otherwise, thank you so much for joining us and have a very nice evening. Thank you.